Thanks for coming to my presentation. My name is Merci, and this is my mentorship presentation, and it's called Off With Their Heads, Fruit Flies and the Basket Gene. All right, so first I would like to thank my parents and my other mentor, Dr. Robinson, and my mentor for this presentation, John Warrick, for coming today. So first I'm going to introduce you to my mentor, Professor Warrick. He is a professor of biology at the University of Richmond, and in his lab we study neurodegenerative diseases, and it's called MJD. I'm going to get into it a little bit more in detail later, but I just wanted to thank him and appreciate him for all the work that he's done
All right.
my mentorship. All right, so my roadmap for this presentation is in the format of a lab report. So who likes lab reports? Okay, we got one. That's So first I'm going to talk to you about my background research up there, and I'm going to tell you about all the concepts that I had to go over and read about to formulate my experiment and my hypothesis. Then I'm going to go into my method, some of the techniques that I did right here. I'm going to talk to you about the techniques and how I got my results. I'm going to talk to you about those. And then I'm going to analyze those so you can see the significance of them and what this research seeks to do. And then I'm going to go into my conclusion and try to synthesize everything that we went over through this presentation. And I'm going to show you how all of that led to my final paper, which is called Examining the Role of Basket Gene in a Drosophila Model of Mikado Zoonosis. All right, so first I want you to get an idea of what I did through my mentorship. So I thought the best way to do that was through a video. So I have a 3-minute video that I'm going to play, and I'm going to narrate throughout it so I can show you some of the important things that I've done throughout my mentorship. So every day when I would come to the university, I would go discuss what the agenda was with Dr. Warrick in his office. And we would just talk about our project and what tasks we were going to get through that day. And these are some other professors at the university. And we're making our way into the lab right here. As you can see, fly flipping is what we do. And right here, you're going to see me sitting at this station where I usually work. And I'm getting my flies out of the incubators where they stay. And I'm turning on the CO2 pump. CO2 is very important when working with flies because it is anesthetize them and makes them sleep while we, do what we have to do with them. All right, I'm getting my lab coat and my fly sucker, which is very helpful in transporting flies from different valves. Now, right here is a technique we call fly flipping. What I'm doing is transporting my crosses of Q27 flies into a clear valve where I'm going to scan them for virgins. Because that's what I did every day. I looked for virgins and I would transport them into my crosses, which I'm going to get into more depth later. So right here, I'm putting some CO2 in the valve, tapping them out, and I'm going to put them on the CO2 bed so I can further analyze them through the microscope. And here's going to be a close-up of that. It says preparing to cut heads. And when I'm done with the flies that I do not need, they go into the fly board where all happy flies go. Alright, and this is Dr. Warrick's office. He has acted as a teacher to me, showing me the retinal staining of the eye and where the aggregates are going to go. I'm freezing heads in this little clip. You won't see the full methods, but I'll just go over that a little bit later as well. And this is the cryostat machine, which is going to be very important in my research. And I'm going to tell you about how I use it to come up with my results. And it took me a long time to master the cryostat because it operates differently due to temperature and humidity was another factor. But I eventually mastered it and got some good results from it. And that's Dr. Warrick with his slides. He also had research that he was doing at the time. And then he started doing some lab chores and washing out valves. You got to keep the lab clean, keep tools clean. And now Dr. Warrick and I, we're analyzing some of the posters that further students have done, and we're making our way to the biological imaging suite, where I got to see my results into fruition, and talk about them with Dr. Warrick, and that's the confocal microscope, which is really important. And this is me working on this recent project that I finished. And this is us in the Dot Wall Center of the Sciences, where I did my research at the University of Richmond. And now you've seen the lab. All right, no applause. Okay, so into my research. So when I say Mikado-Joseph disease, how many of you guys have ever heard of that? One. Oh, two, okay. Mikado-Joseph disease is the neurodegenerative disease that I studied over the course of my project. And it's one disease, but it has different names. The next name is spinocerebellar ataxia type 3, and it's also called SCA3. So one disease, but three different names, but in this presentation, I'm going to refer to it as MJD, or Mikado-Joseph disease. And you're probably like, what? I've never heard of it. Why is this significant? Well, I'm going to tell you that. All right, Mikado-Joseph disease is a human neurodegenerative disease, and it's located in the central nervous system. So right here is a diagram of the central nervous system, which is comprised of the brain and the spinal cord. And MJD is also an autosomal dominant disorder, meaning that it affects families really, really hard. Whereas if one of your parents is affected by the disorder, you have a 50% chance of obtaining the disease as well. So back on this this is showing, pedigree analysis. I'm sorry. It's been a long week. But, this is pedigree analysis. And where you see the parents, the male, the father was affected, and then it goes on into the children, where we see 3 children out of that one family were affected. And then you go through the next generation, and you see 3 more were affected. So this disorder really hits hard in families. So, as I said before, it's a polyglutamine disease where there is an expanded CAG repeat in the gene that causes MJD. And that expanded CAG repeat is also factors into a repeat in the protein where there's a normal repeat of glutamine. So right here in this chart, you see a normal allele. This is a person who does not have the MJD disorder. So in people that have the normal CAG repeat, it's normally like 15 to 20 CAG repeats. However, in people that have the disease allele, it's about 30 to 45 to 50 CAG repeats, which is not good because that's a very toxic protein that can lead to degeneration. All right, and when I say it's a member of the polyglutamine family, that consists of diseases such as Huntington's, and you guys have heard of Huntington's, right? So research with MJD can be very significant to other diseases such as Huntington's and Parkinson's. All right, and so this is the chromosome that we found the MJD gene on. And it's on chromosome 14's long arm, and it's located at row 32, I would say. And that is the genetic locus or genetic location for the MJD disease. All right, and you're probably like, okay, you're talking about the background and the genetic composition of the disease, but you don't really see much about how it really affects people. So this is a person with MJD, and you can see that she has horrible muscle stance where she needs someone to help her up, and she is actually wheelchair bound. She has weak arms and legs, and that is very intricate into the disease where it doesn't really affect the cognitive skills of a person. So the person is still functioning, as we would say, as a normal person. However, it attacks the motor functioning of the individuals where they're essentially trapped inside their own body. They know that is something wrong with them. They just cannot control their body, tell their body to move or do something that they need to do. So symptoms begin around ages 15 to 40. So it has a very wide range of when you can get the symptoms of Mikado-Joseph disease, but they do have the potential to be earlier or later. And other symptoms include weakness in the arms of legs, spasticity, staggeriness, difficulty in speech, difficulty in swallowing. And after they portray the symptoms and get diagnosed with MJD, they have about a 10 to 30-year life expectancy. So this disease lasts a little while longer, so they have to suffer through these symptoms for a lot after they are diagnosed with the disorder. And so when we're studying MJD, and my personal project in particular, we look at this gene called BASKET. And BASKET is the Drosophila homologue of the human gene called Jun amino terminal punase, and it's JUNK. And so this is a very confusing diagram of the JUNK pathway, so I'm not going to expect to analyze everything that's on here. But what I want you to get from this is that JUNK is an evolutionary homologue of the human, well, BASKET is the evolutionary homologue of the human JUNK gene, which is depicted here. So in flies, they have this protein called BASKET, which I studied in my project, that mimics the protein that humans with Makata-Joseph disease have called JUNK. And BASKET regulates a large number of cellular processes, in Drosophila in particular, and autophagy and regulation, which I'm going to get into in the next slide. So autophagy is another process that I study, and it's an evolutionary conserved self-eating process that has been implicated in a number of human neurodegenerative diseases. And in this project in particular, I studied Makata-Joseph disease. And there are multiple types of autophagy, and right here is depicted as macro, micro, and chaperone-mediated autophagy, and they all have this same process where it's the synthesis of a phagophore membrane that goes through nucleation, membrane expansion, fusion, and degradation in the cells. So, you're probably saying flies as a model. Why would you use flies to study such a terrible human neurodegenerative disease? Well, flies as a model are widely used because what we can actually do now is translocate genes from humans into a fly model and create a transgenic model of flies. And we can study pathology a lot faster in flies because currently doing that in humans would be completely unethical, and you have to think about waiting through the generations because it takes longer for humans to have children and pass on genes as compared to flies who have an 11-day life cycle, where from the time that they're born to the time that they reach adulthood. And they also have about 40 to 60 offspring at a time. And what people do not know is that flies have a high degree of homology with humans at a genetic level and a molecular level, where the genes that function in flies are pretty much the same as the genes that function in humans, and many people do not know that. All right, so you're probably saying, how did you do this? Well, the first step into actually performing my research was to set up 2 lines of flies. So I'm going to write this on the board so everyone can keep up with this. The first line of flies that I set up were the Q27s over Q27s, and these were the normal flies. And the second line I set up was the Q84s over Q84s, and these were the disease flies that had the Makata-Joseph disease. I'm going to write it on both sides for everyone to see. All right, so after I set up these 2 lines of flies and grew them and populated them over time, I set up my crosses. Well, here's a picture of that. And I set up my crosses where I crossed flies that had the Q27 over Q27 genotype with flies that had one functioning BASKET allele. And then I crossed that same Q27 over Q27 genotype with flies that had 2 functioning BASKET alleles. Because the whole purpose of this project was to see what effect the BASKET gene played in fruit flies and aggregate formation, which I'm going to go into more detail later.